to all the other students as well. So let's wait for the students to join, then we'll start with our class. Today we are starting off with a new topic in cell biology, that is cell cycle. So in this class we'll be discussing about what cell cycle is, what are the various types, what are the various steps, etc, etc, right? So those of you who are joining new, we have 4 new students in the batch. Those of you who are joining new, please make sure that you are going through the recorded sessions. We have already covered a lot of content in the classes, so make sure that you go ahead with the same. Good morning, Akshay. Okay, so let's begin with our class now, right? So we've already covered 60% of cell biology, right? And we are starting off with cell cycle. So now cell cycle, cancer, and apoptosis, these 3 topics would be covered all together along with the previous year questions. Okay, so let's start with cell cycle. So what do you mean by cell cycle, everyone? Try defining cell cycle according to your understanding. Good morning, Amrita. Good morning, Arindam. Yes, what is cell cycle according to your own understanding? Think about it, everyone, and write down in the chat section. Yes, try defining a cell cycle according to your understanding first, everyone. All organisms begin their life in the form of single cell. That is what that is you're defining a cell. I'm talking about cell cycle, Divya. Cell division, very good. Series of events that occur in the cell for the growth and development, very good. Complete cycling manner by which one cell divides into two and so on. Yes. So what is cell cycle all about? So when we look into the cell cycle, so it is a sequential event in the life of the cell because of which it divides into two, or it divides into its daughter cells, because depending upon mitosis or meiosis we can have 4 cells as well. So depending upon which particular cell are we talking about, that is, there are 2 type of cells in our body. First one, on a broader level, if we see, first one are the somatic cells, and the second one are the gametic cells, right? The gametes via which the gametes are formed. So somatic cells and the gametic cell, depending upon which cell we are talking about, the series of events that a specific cell undergoes in order to make its copies, in order to increase the number of the cells in the body, right? When we look into the cell cycle on a whole, first I'll explain you the entire cell cycle basics via this slide itself, and then we'll go through the presentation. Okay, so listen to it very, very carefully. So when we look into the cell cycle on a whole, there are 2 main steps for the same. The very first step is your interphase, and the second step is your M phase. Now this M phase can be mitotic, it can be meiotic. Okay, let me write down on a separate page. Yeah, so when we look into the cell cycle, in the cell cycle, there are 2 type of cells. The cells could either be somatic, the cells could either be gametic, right? Now when we look into the basic type of steps that occurs in here, so it is a there occurs sequential events in 2 manner. Like there are 2 main phases. First one is your interphase, and the second one is your M phase. Now this M phase can be mitotic phase or it can be the meiotic phase, depending upon whether we are having the division in our somatic cells or in our gametic cell. Right now, when we look into the division or when we look into the cycle that is occurring in our somatic cell, so it is given the name of mitosis, and second one is the division which is occurring in our gametes, which is your meiosis. Right now, when we look into mitosis, so mitosis is equational division. That is, there is one cell, like initially we have the parent cell. This parent cell divides into two daughter cells which are exact copies of each other, of the previous generation. Meiosis is reductional division. Okay, in this, what happens in this, the new type of cell or the new generation of the cells that are formed or that are created, they are not the exact copies. There occurs crossing over event in the complete process. At which particular stage does it occur? We'll understand. But interphase is common to both of them, to either the mitosis or the meiosis. When we look into the interphase, there are three stages again. The three stages are the G1, S, and G2. Right? G1 phase is your, like this complete phase, this interphase is also given the name of preparatory phase as well. Reason being, the cells are getting prepared to be divided. Right now, in the G1 phase, in the S phase, and in the G2 phase, there occurs sequential event in which the DNA replication would be occurring, the cell size would be increasing, and all the doubling of the content would be there. When you look into on a broader level, that cell DNA replication occurs in your S phase. That is, now each of these have their own full form as well. G1 is gap 1, S is synthesis, and G2 is gap 2 phase. Right now, this is about your interphase. There are chances that the cell might exit out of your cell cycle as well, and if the cell exits out of the cell cycle, it is given the name, or it enters into G naught, which is your resting phase, or it is also given the name of quiescent phase. Okay, now next is your M phase. In your M phase, that is mitosis or meiosis. Like if you look into a broader level, like the M phase of any of the division, there are four main stages. Like if you want to bifurcate into sub two stages, one of them would be cytokinesis, second one would be karyokinesis. Right, karyokinesis, karyo is the term related to which particular cell organelle? Everyone think about it. To which particular organelle does the word karyokinesis is uh, related to? Yes, to which organelle this particular word is related to? The nucleus. Very good. So first is your karyokinesis, that is your nucleus content gets divided. Second is your cytokinesis. Cytokinesis is when the entire cytoplasm gets pinched off into two of the types. So karyokinesis and cytokinesis. Cyto is related to your cytoplasm, and kinesis means division of the two. So karyokinesis, then cytokinesis. Now if you look into karyokinesis, there are again four phases. First one is your prophase, then you have metaphase, then you have anaphase, and finally you have the telophase. Right, prophase, metaphase, anaphase, and telophase. This is your entire karyokinesis cycle. After the telophase, then there occurs cytokinesis, that is the final dividing of the cytoplasmic content of the cell. Now, if I talk about the mitosis and the meiosis, in mitosis, this thing would be occurring: the prophase, metaphase, anaphase, telophase, all these things would be occurring. When we look into meiosis, there occurs two of them. So first one is meiosis 1 and second one is meiosis 2. Why is it occurring? Why two of them are occurring? First, because there need to be crossing over. Second, there need to be separation of the sister chromatids as well, right? In the M1, like these four stages would be same. Now, but the difference would be, it would be prophase 1, metaphase 1, anaphase 1, telophase 1. And in metaphase, like meiosis 2 as well, you will be having prophase 2, metaphase 2, anaphase 2 and telophase 2. Okay, difference over here comes in the prophase 1. In the prophase 1 we have 5 stages again: leptotene, zygotene, packetine, diplotene, and dikinesis. In your packetine stage of prophase 1 of meiosis 1 in the meiotic pathway, we have the crossing over occurring. These, whatever, there are various pointers which I have marked on this complete slide. All of these are important. Okay, we'll be going through the complete pathway. Don't worry about that. I'm just giving you an overview regarding the same. Okay, so you have the Packaging stage over where the crossing over occurs, that is over where the DNA recombination and the various types or the various combinations are occurring in over here, because of which there occurs increase or there occurs changes in the daughter cells of your meiotic cell and of your meiotic division and mitotic division. In mitosis, the number of the chromosome, let's say initially is 2n. In the next generation also it would be 2n itself. In meiosis, what happens? Let's say this is one cell, it would be dividing into four separate cells, so all of these would be haploid in nature. So there occurs reductional division. The reduction in the number of the chromosomes is there, or it is seen in case of your reductional division, right? So this is your entire cell cycle basics, that what are the steps and all those things. Just go through it once and then we'll start off with our main segment of the class. Okay, understand, like if you are making your own notes, I would suggest you write this complete thing in your notes in the initial page itself. It would help you out in revising or recalling the entire content within a go. Okay, quickly check this. Understood, everyone? Clear? Very good. Others also, just let me know. 13 of you are there in the class right now. This is the basics of the cell cycle. Now we'll start it, that what is it all about. Okay, very good, let's move forward. Now, what is cell cycle all about? So, when we look into the cell cycle, it is the series of the events that take place in a cell leading to its division and production of two of the daughter cells. So, you have one main cell or the parental cell. This parental cell is dividing or leading to the production of two of its daughter cells. It is a very regulated process, very regulated. Reason being, it's a cell cycle. The number of the cell is increasing in the cell. So if in the body, if there occurs any kind of mishappening, if there occurs any kind of wrong copying or wrong, any process gets dysfunctional, it can lead to very serious diseases in the body as well. Can you write down in the chat section which is the most severe disease which occurs because of malfunctioning in the division of the cells in the body? Everyone, think about it. What is the name of that disease? Cancer, very good. So cancer is what? There occurs dysfunctional or there occurs abnormal division of the cells in the body, which leads to the formation of tumors and other subsequent processes occurs thereafter, right? So it is a highly regulate, the cell cycle is a highly regulated process which is ensuring proper growth, proper development and proper maintenance of the organisms. There are four main stages: G1, S, G2 and M. G1 to S is also called as the preparatory phase. So this complete interphase from G1, S and G2, this is a very long phase, and in addition the mitosis is a very short phase in comparison to your complete interphase. In the examination there are questions asked related to the duration or related to the time that is required, that is being taken up in the cells in order to divide, right? So we'll be having a discussion regarding that as well. When we look into the G1 phase, that is your gap 1 phase, so it is during this phase that your cell is growing in size, it is synthesizing a number of proteins, it is carrying out its normal functionality also. The cell is preparing the content so that in the next phase, in your S phase, the DNA replication can be done. Very, very important. All the proteins that are required for your DNA replication. What is DNA replication in a simple definition? So it is nothing but the division or the copying of your, or we can say doubling of the genetic material, right? The chromosome is getting duplicated. So DNA replication, which we'll be studying in the next unit. So all the proteins that are required in the prokaryotes, in the eukaryotes, different type of proteins are required for the complete DNA replication to occur. So in the gap 1 phase, all these proteins are prepared beforehand so that as and when the cell enters into the S phase, it does not have to wait for the proteins to be synthesized, right? So this is your G1 phase. Then you have the S phase. So S phase is that in this phase, cells DNA is getting replicated. It is resulting in the duplication of the genetic material. Each of the chromosome is duplicated, and after the replication, each chromosome consists of two identical sister chromatids that are held together with the help of a centromere. If you remember, we had a discussion regarding the structure of the centromere also in one of the class where we were discussing about cytoskeleton. So I am expecting that you all remember about the structure of the same. Then you have the G2 phase. So it is during the G2 phase that the cell is continuing to grow and prepares for the cell division. But now the preparation is what? Now the preparation would be the content which is required in your mitosis, right? So G2 phase, in this phase the cell continues to grow and prepare for the cell division. It synthesizes the necessary proteins and the organelles that are required for the cell division. The cell checks for the errors in the DNA replication and it initiates the repair mechanism if it is required. Now you have to remember this about the complete thing, that is, there this complete cell cycle is a very regulated manner at each of the junction, whether it is the cell entering from the G1 to the S phase, from the S to the G2 phase, from the G2 or to the M phase. So at each of these steps we have regulations prepared done, and these regulations are called as, or they are abbreviated as, the checkpoints, and these checkpoints are taken care with the help of cyclins and CDKs. We'll be discussing about all of these all things are very very important. So because obviously it's a cell cycle, or the complete cell is getting divided, that means what? That means it need to be very much regulated. There need to be a very proper regulation in the entire process. So these checkpoints are helping in the same. Okay, now you have the M phase, that is your mitotic phase. Arre, yeah. So you have the M phase or the mitotic phase. This is the main phase of your cell division. It is consisting of a number of other sub phases as well: prophase, metaphase, anaphase, telophase. Prophase is what? It is the chromatin getting condensed, the duplicated chromosomes becomes visible, and the nuclear envelope disintegrates. Mitotic spindle begin to form. Mitotic spindle, to which particular cytoplasmic cytoskeleton content is it related to? Yes, write down in the chat section. We have discussed about this. Mitotic spindles are formed with the help of which of the cytoskeletal component? Think about it. I'm talking about mitotic spindles are formed by which of them? Centrosome is not over, like centrosome will not be forming the mitotic spindle, na? Centrosome would help in the alignment of the same. I'm talking about the cytoplasmic or the cytoskeleton content. Microtubules, yes, via the help of microtubules it would be formed. If you look into the structure over here, the centrosome would be there, and the microtubules would be, or the mitotic spindles will be formed, or they will be assembled with the help of microtubules, tubulin protein. If you remember, we have discussed all this thing. That's why I was saying that after you are like on the other side of 25 classes, you start to forget the previous things, because initially it's like 4 classes, 5 classes, but now you have 28 classes in hands. So you'll start to forget things, and it's just unit number 2. As and when we'll reach unit number 13, you'll see more than 150 classes would be there. So please make sure that you are revising very very properly. Okay, yesterday the scorecard came and many students had this problem that they did not revise properly. So please make sure that you are revising the previous content very effectively. Then it is metaphase. So metaphase is that the spindle fibers attached to the centromeres of the chromosome, aligning the chromosome at the equator, also known as metaphasic plate. I'll show you the diagrams also, wait for it. Then you have anaphase. So the spindle fibers shortens separating the sister chromatids of each of the chromosomes, and the separated chromatids move towards the opposite poles. Let's say this is your chromosome. Sorry, very bad figure. Let's say this is your chromosome. So this chromosome initially gets aligned on the equator at your metaphase time. In the anaphase, the mitotic spindles start to shorten up and the sister chromatid starts to move towards the opposite poles. That means the arms, like two of these would be moving towards the opposite pole, and as a result, the separation of the sister chromatids would be there. Okay, do not get confused, wait for it, you'll get the complete, like with the help of diagram I'll explain. Just understand the entire thing right now. Next, the telophase, the separated chromosome. Now the sister chromatids are onto the opposite poles. There need to be decondensation of the chromosome as well. It need to convert back into the chromatin fibers, right? So this would be occurring at the opposite poles. Cell undergo cytokinesis, the division of the cytoplasm, resulting in the formation of two of the daughter cells, right? I've explained this particular thing just now. So same thing is written. Then you have, after cytokinesis, the two daughter cell enters the G1 phase and the cell cycle starts again. So this is a complete cycle. As and when the daughter cells are formed, now the next cycle would initiate and the work would keep on getting done, right? So this is the entire cell cycle. So look over here. There is occurring cell growth in the G1 phase, DNA replication in S phase, preparation for mitosis in the G2 phase, and then the complete cell division is occurring in your M phase, that is your prophase, metaphase, anaphase, telophase, and finally the cytokinesis. These four steps can also be called as, or there over here there is occurring division of your nucleus. So it is also, can also be abbreviated or can also be termed as karyokinesis. Okay, so this is the entire cell cycle. Now when you look into meiosis, the difference between the mitosis and the meiosis, so let's understand about meiosis now, and then you'll be able to get the entire difference between the two. Or before this, look into this particular diagram. So look over here, you have mitosis. In mitosis, what is happening in mitosis? In the interphase, 2n. In the prophase, you will be having 4n, because in the initial G1 phase, from G1 to S, the division or the duplication of the DNA would be there. So in the prophase, you have 4. Metaphase, these 4 align on the equator. Anaphase, they start to move towards the opposite pole. Telophase, they are on the opposite pole, and then there occurs condensation of the content, and then you have the daughter cell. So initially interphase 2n, daughter cells also 2n. So it is equational division, right? Then you have meiosis. Meiosis, initially 2n. Over here, S phase would have been taken place. So in the prophase, 4n. Metaphase, anaphase, telophase. So till this particular pointer, and over here same thing is happening. The only difference is in the prophase 1 there would be DNA recombination occurring, there would be crossing over occurring, because of which these are not the exact copies of your initial parent. In the prophase 2, these would be working in individual cells, like in individual area these would be working in. So in the metaphase, the recombined or the crossed over content would be getting aligned, then segregation would be there, and finally the product of meiosis would be having half number of chromosome in comparison to your in comparison to your parent. And as a result of this, and as a result of DNA recombination also, this would also be having variation in it. Okay, so remember about this interphase initially, and then in final one you have the reductional division getting done. Out of the two, which one would lead to variation or which one would lead to diversity among the organisms? Everyone, think about it and write down in the chat section. Out of mitosis and meiosis, which one would be leading to more variations? Very good. It would be meiosis. Which in which particular step do we have the DNA recombination or the crossing over occurring? Yes. At which particular step do we have the DNA recombination occurring in the meiosis? Write down the exact step. Let me see how many of you have listened to the thing. Yes, it is your Packington stage of prophase 1. Very good. Remember about this particular thing. Let's move forward now. Right. Now we'll be understanding about the meiosis 1 and meiosis 2. So meiosis is what? It is a specialized form of cell division itself. The only difference is that it is occurring in your gametic cells, because over here there need to be changes, there need to be much more variations, differences should be there in the content. So meiosis is occurring in here. It is a specialized form of cell division that occurs in the reproductive cells, and it is involved in the sexual reproduction. Okay, it reduces the number of chromosomes by half. How does it actually does? We have just discussed, and it results in the formation of what? It results in the formation of haploid cells or the gametes, in which half the number of chromosomes compared to the parent cell. So in the normal cell, in the first cell we have the 2n, and in the final cell you have the products is n. So complete reduction, like exact half number is there now. Now there is meiosis 1 and meiosis 2. Meiosis 1, each of these is containing prophase, metaphase, anaphase, telophase, and the cytokinesis. So prophase 1 is prophase 1, sorry, in the prophase 1 you have the chromosome getting condensed, you have the homologous chromosomes that pair up in a process called as synapses. Then you have crossing over, exchange of genetic material between the homologous chromosomes is occurring. Then you have metaphase. So metaphase is homologous chromosomes aligning at the metaphasic plate. Anaphase, they are moving towards opposite pole. Telophase, they are getting condensed over there. Cytokinesis would be following, resulting in two of the haploid cells with duplicated chromosome. Now, in the meiosis 2, this particular content would be getting reduced into half. So prophase 2 is chromosome recondenses, nuclear content, the nuclear envelope breaks down again. After that, metaphase 2, chromosome align at the metaphasic plate. Anaphase 2, sister chromatids would be getting separated. Telophase, same thing. The only difference over here in the meiosis 1 and meiosis 2 is that in meiosis 1 you have the homologous chromosome, in meiosis 2 you have the recontents, like the duplicated content which was there, that is getting separated into half of the two. Okay, so the genetic variability in the sexually reproducing organism arises from what? It arises from independent assortment of the chromosome. What do you mean by independent assortment, everyone? Think about it. What is the meaning of the word independent assortment? Yes. What is the meaning of the word independent assortment? It means that the inheritance of one gene is unaffected by other. Very good. So independent assortment, this is a rule, or this was the genetic rule that was given by respected scientist Mendel. We'll be studying about that in unit number 8. So in that, the one of the rule that was given was, or one of the law that was given by Mendel was that the genes which are getting inherited to the next generation, they are independently assorting. They do not get affected, or they do not depend on any other gene for the same matter. Okay, so over here itself the same thing is written, that the genetic variability is in sexually reproducing organisms from the independent assortment of the chromosome from during meiosis 1 and exchange of the genetic material through the crossing over. The resulting gametes have a unique combination of genetic information, because of which the content is, or the new generation which gets formed is having variation in comparison to their parents, right? So look over here, we'll start from the G1 phase, that is from this phase. So in the G1 phase, what is happening? There is in division, or the cell is starting to grow over here. Then you have, oh, there. This is about regulation. Wait, yeah, this one is better. Look over here. I hope I are able to look into it properly. So first one is the gap 1 phase, where the cell is growing. Then you have the S phase, the DNA synthesis is occurring. The cell would duplicate its content, and the centrosome would be duplicating. Then you have the gap 2, cell is growing to a much more broader level. Prophase 1. Now these four stages are like, at which stage what particular exact thing is happening. Understand it. Yeah. So you have the prophase. In this, the chromatin condenses into chromosomes and the nucleolus is disappearing. Pro-metaphase, like when we look into the metaphase, in some of the books there is mentioning of pro-metaphase plate, or the pro-metaphase uh, step as well. It is not that you have to mark it every time that there are 5 stages. No, there are 4 stages only. But some scientists consider that there is a pro metaphase plate as a pro metaphase step as well, in which the cell is just cross verifying that is everything ready for the metaphasic side or not, or for the metaphasic step or not, because it's a very very important step. The chromosome would be getting aligned on the metaphasic plate, and from this step itself there would be separation, or the segregation to the opposite poles, and that will be getting condensed and forming the new daughter cell. So that is the reason why this step is very important, and hence a pro metaphase step is recommended, or it is suggested by few of the scientists. So in this, what is happening? The nuclear membrane breaking down, kinetochore microtubules invade the nuclear space and attach to the kinetochores. Polar microtubules push against each other, moving the centrosomes apart. Because of the centrosomes moving apart, the alignment would be proper. Because from after that, only the microtubules would be, or the spindle fibers would be getting attached to our kinetochores, and the work would be getting done. Then you have anaphase. The chromosomes break at the centrosomes and they move towards the opposite poles. Telophase and cytokinesis. So the nuclear membrane reforms, nucleoli reappears, chromosome unwind to the chromatin, and finally the division into the two daughter cells is done with the help of your cytokinesis. Okay, so this is your entire cell cycle. Now you have two of the homeworks which you have to find, and you have to post it in the chat section, in the class, in the group. First thing is, you have to find out the duration of the phases, that at what is the duration of which particular phase, because in the examination you can get it. Second thing is, you have to find out the DNA content. How does it increase, how does it decrease after separation after segregation, or how does it increase at which particular stage. Okay, so the DNA content and the chromosome content also. Okay, this is your second homework. You have to find out the chromosome content and the DNA content in mitosis also and in meiosis also. Very, very important. And you'll be posting it on the group before the evening class. Okay, in the evening we'll be having the regulation discussed. So before the evening class, you have to search these two things. Okay, now let's move forward. These are the two main homeworks which you have to find out. Make sure that you are doing it and posting it on the group. Now, same thing is over here, just a new way of representation. Prophase, metaphase, anaphase, telophase, and the cytokinesis. Exact similar thing is over written over here, just for your more better understanding. So, condensing chromosomes with two chromatids held together at the kinetochores. Prometaphase, the complete spindle fibers, sorry, the centrosomes move towards the opposite poles and all the things are checked properly. Metaphase, this align at the equator plate or equatorial plate. Anaphase, they move towards the opposite poles, increasing the separation of the poles. Telophase, the reformation of the individual chromosome occur on the opposite poles. And cytokinesis, the cytoplasmic content is getting divided into the two. Okay, so this is your entire mitosis. So mitosis versus meiosis, we have discussed this. So again, just a new way. Prophase, metaphase, anaphase, end result. In the meiosis, prophase 1, metaphase 1, anaphase 1, telophase 1, and the meiosis 2. So meiosis 2 is somewhat related, or this almost similar to your mitosis itself. It is just the meiosis 1 in which the prophase 1 is very elaborated and you have a number of different things getting done, like DNA recombination, crossing over, and all those things. So otherwise, in the meiosis 2, exact same thing is occurring what happens in your mitosis, right? Same, the chromosomes would be getting decondensed, then the move alignment on equator and all those things, similar thing would be there. Only difference is in your meiosis 1, because for the variations to occur in the cells, this complete process occurs. Okay, so this is your mitosis and meiosis. This is done. This is regulation. Before this, we'll be solving out few questions. Is everything clear to everyone till here related to your complete cell cycle? I've tried to make it as elaborated as possible. If it is clear, we'll move forward. And this particular topic is very important, even if you are not just preparing for CSIs and other exams, like your state PCS or any other exam. For that also, this particular topic is, or this complete content would help. Okay, almost in every exam we have the similar syllabus, only the pattern of the exam and the demand of the exam changes, otherwise the content is almost similar. Clear everyone? 9 students in the class. Okay, let's move forward. So I'll give you a few questions now, and then in the evening we'll start off with the regulation of the cell cycle. Okay, try solving out these questions. Okay, give it a try to this question, everyone. Think about it. I'm making the chat private. Think about it and mark the answer. quick. Okay, let's see. The key determinant for the plane of cytokinesis. That means you have this cell which is undergoing division and everything. It is asking plane of cytokinesis would be defined by which of them? Yes. Now think about it. Now answers of many students would change. Plane of cytokinesis would be defined by which of the four? So answer would be option number B. That is the central spindle. Right, there would be a spindle over here as well, which would be pinching it off. And if you look into the other three options, it is nowhere related to your cytokinesis. Pre-prophase band, not possible. Chromosomes, they have already condensed over here. Centrioles, they are to the opposite poles. How can these three work? Answer would be central spindle. Clear, everyone? One, two, three of you got the answer correct, and one of you made your answer correct yourself, incorrect yourself, first marking the correct one and then changing the option. So understand this properly, that in the examination you have to keep your eyes very very open via understanding that, like, if you don't know the correct answer, try to find out the not possible statements and then you'll find the correct answer yourself. Next question. This is a June 2022 question. The question is individual chromosomes can be seen clearly in which phase of the cell cycle? Latest year question, like September 2022, a batch question. Individual chromosomes are clearly seen during which phase of the cell cycle? Think about it. Done, everyone? Option number D. See over here, they have the CSIR should have written meta phase over here, but they have written it in a general manner of M phase. Like, students can take this as M phase also and as a meta phase also. Okay, so individual chromosomes are clearly seen during the meta phase itself. Okay, specifically meta phase, but because over here they have not mentioned what do we have to consider it, so remember that it is the meta phase of the mitotic phase. One more question. Oh, this one you won't be able to do. This is regulation. Yeah, you can do this, I guess. The mammalian oocyte prior to the sperm entry is arrested in which of the stage of the cell cycle? So give it a try to this. It's a complete new information which you can take up from this question. So take your time, get the answer for this. Think about it. What could be the answer? There is no two, either A or B. I won't be taking the answer as A or B. Either of them. You should write either of the option. Okay, Arindam, let's check. Okay. So, question is the mammalian oocyte prior to the sperm entry is arrested in which stage of the cell cycle? So, it is your metaphase of the meiosis 2, because after that stage, Divya, you have marked it answer B. Okay, answer is option number D. That is, it is the metaphase of the meiosis 2 in which the mammalian oocyte gets arrested, or mammalian oocyte prior to the sperm entry gets arrested over here. Okay, so this is the answer for this particular question. Metaphase of the meiosis 2. Remember this particular thing as a fact. Okay, give you can give it a try. Okay, give it a try to this question as well, everyone. Think about it. Question is when a yeah, when the cells enter the mitosis, their existing array of the cytoplasmic microtubules has to be rapidly broken down and replaced with the mitotic spindles, which pulls the chromosomes to the opposite cells. There's an enzyme catenin which is activated by the onset of mitosis and it chops the microtubules. So you have to find out what is the possible fate of the microtubule fragments that is created by catenin. If you put in your basic knowledge to it, that because microtubules has to be used in the recurring cycles, so which of the following should be the fate of the microtubule? Think about it, try to put in your basic or very general knowledge into this. Let's see. Polymerize. Very, very simple, right? Because it needs to be used in the next cycle also. So it would just get depolymerized, and as and when the next cycle initiate, it would get polymerized again. Getting aggregated together, not an option. Degraded, no, because why do a cell have to use such a important content, or why do a cell want to make such a content again and again by just wasting energy? So degradation, no. Translocation, why will it get translocated? Because it would have to be used up in the next cycle very soon, so it would not be getting translocated also. Very, very simple answer is option number A, that is depolymerization would be there, and as and when the cell need it again, it would start to repolymerize it and use it in the next cycle, right? Simple question. And this was the 4 marker in the CSIR. It was a 4 marker question. One more question, give it a try to this. Very important, very easy question again. Which of the following statement about meiosis is not true? 4 options. Whatever we have discussed, according to that, you will easily able to solve this question. Mark the answer for this as well, everyone. Done, everyone? Statement about meiosis is not true. See, if you look into the first and the second option, these two are contradictory statements, right? Now, either the first one is correct or the second one is correct. Kinetochore of the sister chromatid move towards the opposite pole in meiosis 1, and then attached to the opposite poles in meiosis 1, and kinetochore of the sister chromatid attached to the opposite pole in meiosis 2. Answer is first one. The reason being, the sister chromatids move or attach to the opposite pole in meiosis 2 and not in meiosis 1. Which chromatids get organized in the case of your meiosis 1? Write down in the chat section which particular chromatids arrange or attach to the opposite pole in meiosis 1. Think about it. Yes, homologous chromosome. Very good. So, answer will be option number 1 as the incorrect answer. Not true. Except for this, second, third, and fourth are correct. Okay, very, very simple, very easy. See, we have done the basics also, and with that, we have done the questions as well. So, I hope all these things were clear to you. Right now, in the evening, we'll be having a discussion regarding the cell regulation. Okay, so we'll be having a discussion regarding the cell cycle regulation. So, this was about for morning class. Be there in the evening, 6 o'clock. All the best. Good luck, everyone. Study well and get your homework done also. Okay, bye, everyone. Take care.